everyone. Thanks for being here today. I'm Molly Hurst. I'm from the Cortez Ortiz lab. And I am honored to present to you Erica Larson. She started her academic career over at Western Washington University, where she got her bachelor's degree in biology and chemistry. She then, after a few years, earned her PhD at Cornell University under the guidance of the late Dr. Rick Harrison. And there she studied the dynamics and patterns of introgression and reproductive isolation in a mosaic field cricket hybrid zone. And after that, she went to the University of Montana, where she worked with Dean Good as a postdoc. And there she started studying the genetics of hybrid male sterility in the house mouse hybrid zone. And then in 2017, Erica started her position as associate professor in the Department of Biological Sciences at the University of Denver in Colorado. And over the years, she has published a lot of high-profile papers in several really well-known journals, such as Genetics, Nature, Ecology, and Evolution. She's also received a lot of awards for her research. She has also mentored a lot of undergraduate students, and she's currently mentoring 2 PhD students. She also has a really outstanding record of outreach. And another really interesting thing that I saw on her CV was that Erica has dabbled in wildland firefighting, which I thought was really interesting, but I guess it's a very common thing to do over on the West Coast. So today, Erica is going to tell us about the evolution of polymorphic hybrid male sterility in the house mouse. And with that, I'll leave it to Erica.
So can you hear me okay? This is working? Okay, since Molly brought it up, I was a wildland firefighter. That is how I earned my way through undergraduate degree, and like with fires on all of our mines in California right now, it's very real if you've seen those. It was a very interesting experience as an undergrad. And in terms of the outreach, I think the reason I do it is because if it wasn't for that kind of outreach, I definitely wouldn't be here. The possibility to work on this hybrid zone that I'm going to start my talk with is really what got me interested, not just in the field of evolutionary biology and going to graduate school, but like really in my core research focus, which is hybrid zones. And it was part of working on this system and getting the opportunity to do undergraduate research system instead of firefighting that is why I'm here. So it's a pitch for those NSF broader impacts. So basically, I fell in love with speciation in hybrid zones working on these, I think, very charismatic little green and blue leaf beetles. These guys are specialists on this host plant called dogbane. It's similar to milkweed, which you guys are probably more familiar with. And this plant produces these toxins, these cardinalides, that the insects sequester, and they make the insects toxic, right? So they're aposematic. They have this bright coloration. They hang out on the tips of the plants, and nobody wants to eat them because they're peppery and cause heart attacks. And so these 2 species, they're very cute, and where they come into contact in eastern Washington, they form a hybrid zone. And I'm from eastern Washington, so this was sort of what got me started in undergraduate research. And where these 2 species come into contact, you can walk into the area where their ranges overlap, and you can observe them just hanging out on the leaf of their host plants, and you can see them mating like this on the host plants. And these 2 species form this intermediate purple hybrid. And when I saw this as an undergrad, I was just in love, because this is like one of the coolest things about hybrid zones, that they are places where you can actually walk across the range of the edge of one species range into another, and you can observe these spatial patterns of changes in morphology, ecology, behavior, and you can see evolutionary processes such as hybridization happening right in front of you. And you can observe the outcomes of those interactions through the hybrids and then the impacts those hybrids have in terms of fitness and their interaction with the landscape and other organisms. So because of this, evolutionary biologists and naturalists, we have always been pretty obsessed with hybrid zones, and I think they're just fascinating. And it's this spatial variation that we're interested in, how you can see these transition in traits through space and time. And so this is just an example of some hybrid zones that I could fit on a single slide. I've worked on a few of these. Others in the room have also worked on quite a few of these too. And these are systems that have really motivated and helped our understanding of the nature of species boundaries and what we mean when we say that these are two independent evolutionary units that we call species. And as my advisor, Rick Harrison, would say, hybrid zones are windows on evolutionary process. But as he pointed out, these windows open onto many different landscapes. And I think this is the struggle in speciation, is that there are many, many, many landscapes. And it seems like each one of these systems tells us a different story about the origin of species. And the struggle is to understand the commonalities or the themes, if there's rules to how species form and maintain themselves in the face of like secondary contact. And so I think in speciation, maybe one of the places where we've made the most progress towards understanding common themes or rules is in the study of these postzygotic barriers. So these are barriers to hybridization that happen after a zygote is formed, so after mating and after fertilization. And these are traits such as like hybrid inviability or hybrid sterility that can make hybrids less fit. They can also be external things like a hybrid's less fit in the environment. For today's talk, I'm mostly talking about intrinsic postzygotic barriers, just for any of you who care about that difference. And so this is just an example of some of those hybrid zones, a subset from the last slide, where we know postzygotic barriers are important to how these species actually maintain their identities in secondary contact. And postzygotic barriers are also they're the trait that we sort of know the most about the genetic basis of species genes or traits that keep species apart. And that's because, as Darwin pointed out, there really can't be selection for hybrid sterility. You can't envision two populations having some selection for traits that would cause an intermediate to be less fit, because that would involve an individual like going through a fitness valley. And so there's this model that we have for understanding how these sorts of postzygotic traits can evolve, and that's their interactions between multiple alleles, different genes that interact negatively with each other. And so the idea is that the ancestral population can become divided, like geographically, for instance. And in these isolated populations, these populations can derive new alleles that become fixed eventually. And then if they were to come into secondary contact, this A allele, big A allele, and big A allele and big B allele have never before seen each other. And when tested in an F1 hybrid background, they don't work, and it causes some sort of inviability or infertility. So that's sort of the model for how we think about the evolution of these postzygotic traits. Now, we know the most about this genetic basis for speciation. There has been a ton of work, especially in Drosophila, also in house mice and other systems, that have actually been able to identify genes that we know are involved in Dobzhansky-Muller incompatibilities. And so we know a lot here, but if there's one takeaway I want you to get from this talk is no matter how much, we still don't know so much. So even in these best studied cases, it is way more complicated than this simple model. For instance, there's been very few systems where we have carefully looked at these sorts of incompatibilities in natural populations and things that are actually interacting in hybrid zones. A lot of this work has been done doing crosses in the lab with lines of organisms. Also, it's not so simple. Like if you kind of start thinking about this, why does it have to become fixed? There could be polymorphism at these loci. There could be polymorphism in the ancestral population for these alleles that could later cause incompatibilities. There can be polymorphism in each lineage. And then together what you get is a much more complex genetic architecture for what we're modeling as sort of a simple trait here. And so there's a lot of work to be done in sort of understanding these traits in real populations and real organisms. And I think an excellent example, we know from studies of hybrid zones that there is variation in how reproductively isolated lineages are from each other depending on where you sample individuals. So an individual from population A might work fine with the other species, but you sample one from some other part of the range and they don't work. And so there is evidence in some of these systems where there's variation in how reproductively isolated these lineages are. We also know from crossing experiments, like a lot of different lines, like Arabidopsis and things that don't necessarily form hybrid zones, but that there is variation for these Dobzhansky-Mueller incompatibilities within species. So the thing is we know this variation exists, but we don't know a lot about where it comes from, how it's maintained in a population, and then what that actually means in terms of reducing gene flow in an actual hybrid zone. And so that's where I think that we need a lot of future work. This is really exemplified in the house mouse system. So these two mice, they're very closely related. They're very morphologically similar. I just happened to pick one with a little white cap on his head. They don't all look like that. If you pick up two, only one person in this room might be able to tell them apart, but like they're really similar. And they're ecologically similar. They eat and do the same kinds of things. But where these two species come into contact in Central Europe, they form a stable hybrid zone. So they're not just merging in and forming a single species. They're distinct. And perhaps the primary trait that is keeping them isolated is the sterility of F1 hybrid males, or of hybrid males. And so where these two species come into contact, you find hybrids, but when you cross them in the lab, you find that those males, hybrid males, tend to be sterile. And so if you take a domesticus female and a musculus male and you make a hybrid, the females are always fertile, almost always fertile, and the males can be sterile depending on which population you take to cross. So there's polymorphism there for that sterility when it's a domesticus mother. When it's a musculus mother, they're almost always sterile. So here, whenever you have a musculus female with a domesticus male, you almost always get a sterile male. And again, the females in this cross are also fertile. So these sterile males, they have small testes, low sperm counts, and often abnormal sperm morphology. And so what I want to talk to you about today is I'm going to tell you about some of the genetic pointers really not working. Should switch pointers. Do you have the other one? No, it's gone. We'll use, yeah. Okay, no pointer, that's okay. I can wave my arms. So in this cross, perfect. Okay, sorry for the pause. My battery must be dying. It's this one? Perfect. So in this cross, I'm going to get to, by the end of the talk, focusing on what we think are some of the mechanisms that maintain the polymorphic sterility that we see in this cross direction. But to get to that point, I first want to talk about this cross direction, this one where it's usually sterile. I want to tell you a little bit about the basis of that sterility, and then later in the talk, I'm going to dive into this polymorphic side of things and make it more complicated. So just to begin with, straight off thinking about this, the fact that females are fertile, and you see this really strong asymmetry in the direction of sterility. So they're always sterile in this direction. That right there suggests that there's something really important about the musculus X chromosome in this hybrid sterility because it's asymmetric. And so this is consistent with Haldane. This is consistent with Haldane's rule. So this observation that when you have a problem with a hybrid and it's limited to one sex, that it's almost always the heterogametic sex. And so in this case, you have X, Y are males, and so it's the XY organisms, that are individuals that are sterile, where the XX females are fine. If this was birds, you would see this observation, ZW females would tend to be sterile and males would be fine. And so that's consistent with this, and again, this suggests a large role for the X chromosome. There's lots of other reasons why we think the X chromosome has a large effect in speciation. This is particularly true for the evolution of hybrid male sterility. So we know from other studies of hybrid zones, particularly Priscilla Tucker's work was really revolutionized, some of this early work in the hybrid zones, looking at patterns of gene flow for the X and the Y chromosome versus the autosomes. You can see that the X chromosome here on the top has restricted introgression relative to the Y chromosome, mitochondrial DNA, and I'm not showing you, but autosomes also show more gradual transitions, more gene flow between the two mouse species, where the X, you see these really steep climbs that suggest there's restricted introgression of the X chromosome. So we know from studies of hybrid zones that this suggests there's something about the X that keeps it from being exchanged between the two species. We also know from many mapping studies that both in the lab and with mice from the wild, that hybrid sterility genes tend to be localized on the X. So this is just an example of a QTL mapping study done with hybrid zone mice, and you can see that sterility loci that are associated with like abnormal testis expression and low testis weights are associated with the X chromosome, and also a region on chromosome 17 where there's this gene called PRDM9. And I'm going to get back to this gene later, so just remember that there seems to be this interaction between the X and this autosomal locus PRDM9. So we know from hybrid zone studies and laboratory crosses that sterility seems to be associated with the musculus X chromosome. And there are a lot of different reasons why, technical difficulties. There are a lot of different reasons why you might expect to see the more rapid evolution of hybrid sterility on the X chromosome. So sex chromosomes, because of the way they're inherited, you can have more rapid evolution of genes that are linked to the sex chromosomes. There's predictions that sex-biased genes that might be important in things like sterility could accumulate more quickly on sex chromosomes. There's also arguments for whether or not you can have conflict between like the X and Y chromosome that could lead to more rapid evolution. There's also this theory that there's something about the way the sex chromosomes are handled during development of male gametes that could contribute to hybrid sterility. And so this last hypothesis is one I want to focus the first part of the talk on. And this is the idea that the X chromosome expression is disrupted during spermatogenesis. And so I'm going to walk you through what I mean by this. So this is just a cross-section of a seminiferous tubule. In case it's been a while since you were in intro bio and had to learn about meiosis. Meiosis starts with basically you get proceeding outward on the outside of the tubule, inward to where you get mature sperm released in the middle. And it starts with a round of mitotic divisions, and then these cells, these spermatogonia, enter into meiosis, where you have chromosomes that are pairing up. Homologs are recombining, you get recombination, and then there's 2 rounds of cell division to ultimately produce 4 haploid gametes. And then after that, you actually undergo really dramatic differentiation of the sperm, and this is where you get really interesting sperm morphologies that tend to be variable among species developed during this spermiogenesis phase that happens after meiosis. And so during this developmental timeline, the autosomes, there's a lot of gene expression happening. So the autosomes are being really actively expressed, and there's different genes being turned on across this developmental timeline. We know a lot about which genes and where from humans and mice. And what's really interesting is the sex chromosomes are different. So the X chromosome and the Y chromosome are inactivated in meiosis right after the homologs pair and before you continue on through those cell divisions. So you get this meiotic inactivation of the sex chromosomes. And then the X chromosome remains inactivated for the rest of meiosis, and it's again transcriptionally repressed in this post-meiotic developmental stage. So basically at this stage, the X is like turned down. And so this pattern is really interesting and relevant to what I'm talking about, because if you disrupt this process of turning the X chromosome off, you get sterile males. So you get males that look just like our hybrids. They have low testis weight, abnormal sperm morphology, low sperm counts. All of the hybrid sterility syndrome that we see in a sterile F1 male, you'll see in a normal male if you turn on the X chromosome at this point in development when it should be normally turned off. So this suggests there's something about the X chromosome being expressed during the production of male gametes that somehow screws up spermatogenesis, that makes spermatogenesis halt or fail. And so this is a really interesting hypothesis that's been around for a long time, that this could be a major mechanism contributing to hybrid sterility in a lot of different taxa, at least XY taxa, that have this kind of X inactivation, which is, all of mammals. And so we wanted to test this hypothesis, and it seems pretty straightforward. You would want to take a hybrid male that is sterile and a fertile hybrid male, and you just want to see, is the X on or off, right? Sounds simple. But, and then you could see whether or not there's a role for X regulation in hybrid male sterility. But the problem is it's kind of hard to just compare the gene expression in sterile and fertile males. And that's because testes, as we've just seen, they're really complex tissues. There's a lot of different cell types, and each of these cell types are they're expressing different things. So the X chromosome's turned on early, but then it's turned off, but then it's kind of on again. And so if you just compare two individuals, are you comparing differences in cell composition or gene expression? And so the issue between comparing testis expression between a sterile and fertile male is a sterile male is going to have fewer of those late spermatogenesis cell types than a fertile male. And so they have totally different cell compositions. And so what we wanted to do was to be able to look at this gene expression along this developmental timeline. And so to be able to do this, here's just a picture of what I was telling you, that these males have fewer of those late spermatogenic cell types. So you see these missing versus the complete meiotic complement there in the seminiferous tubules. So what we wanted to do was be able to look at gene expression along this developmental timeline. So to do this, we took testes and we suspended them so that we could then sort the cells based on their overall size and how condensed the chromosomes are during meiosis. So during meiosis, remember, cells are going to start big and then they're going to divide and get smaller. And chromosomes are going to start out loose and then they get more condensed and then they loosen up again. And so these are traits that change across this developmental timeline. And so you can use flow cytometry to basically sort cells based on their size and how condensed those chromosomes are. And what that can give you is enriched cell populations across this developmental timeline. And then we can look at patterns of expression in each of those different cell populations. And so that is what we did. Here I'm just showing you, this is the X chromosome in fertile males. These are just musculus and domesticus normal males. And each individual column is a gene, and each row is a different individual. And so here's patterns of expression on the X with low expression plotted in white and high expression plotted in red. And you can just see a lot of the X chromosomes turned on. Things look good. We isolated cells for these mitotic cells, these spermatogonia. We isolated early meiotic cells before the X chromosome is inactivated. Patterns look pretty similar between those two developmental time points. Here you can see the X chromosome. Well, you can see a little bit. It's almost completely inactivated in fertile males. So it's actually a little more dramatic on this screen than it is if you look at the paper. It's a little washed out here. But that's great for my point. You can see the X chromosome, it's turned off, right, in a fertile male. What little bits of red you see here are very likely downstream undegraded transcripts from this earlier developmental time point, because see how they correspond to really highly expressed genes early on. And so this shows that we have really good developmental timeline data. We can confirm that by these X chromosome patterns. And then if we look late in spermatogenesis, here we see the X chromosome is reactivated for part of the genes that weren't turned on earlier are turned on here, and then genes that were turned on earlier are silenced. And so here you see this really dynamic regulation of the X, where there's some genes that are probably specific to this stage and might be critical in this development of mature sperm and where some of that differences in sperm morphology come from. So we used this data set, and we did the same thing for our F1 hybrids. So now I'm going to show you what those data look like. So here I'm plotting each one of these different black bars is a chromosome, and you have gene expression being plotted above or below the bar depending on whether there's higher expression in the fertile or the sterile hybrid. And so here what you can see is there are very few differentially expressed genes just along the autosomes. This makes sense. These are F1s. They have the same autosomal component. They have one copy of each chromosome from each parent. But the X chromosome in the sterile hybrid is that musculus X chromosome. And you can see that musculus X chromosome is massively overexpressed relative to that fertile hybrid. And that's also true if you compare that F1 to its parent, to a musculus X chromosome in a musculus. So even early on, we see this really big overexpression of genes on the X chromosome. We don't know if this is linked to a phenotype, but it suggests that even early in development, there's already some problems with misregulating the X during spermatogenesis. Here is where we expected to see the X chromosome inactivated in a fertile male, and that's exactly what we see. The fertile males, there's no gene expression. But in the sterile males, you see this massive overexpression of X chromosome. Almost every gene on the X is turned on in a fertile male, and it's turned up. So basically, that's a really great solid evidence of this hypothesis that disruption of that X inactivation could be contributing to sterility at this stage. So, but why? What causes it? So when we were doing this study, there was a paper that came out that gave a really strong model for how this could work. And this is going back to that gene I told you about on chromosome 17, PRDM9. So PRDM9 is a gene that is present in a lot of vertebrates, and it controls for where recombination happens during meiosis. So it's a gene that basically binds to a chromosome and determines where those double-stranded breaks are happening that are allowing the homologs to pair and crossing over to take place. And PRDM9, I won't get into why, but basically it evolves really rapidly. So PRDM9 and its binding sites turn over quickly in vertebrates. And one of the consequences is that between two subspecies of closely related mice, you can get divergent PRDM9 binding sites. And so then what happens is if you make an F1 and PRDM9 likes to bind to the original binding site and not the divergent one, you get this asymmetric binding. And when you get asymmetric binding, what the cell does, if asymmetric binding, you don't get proper crossing over, what the cell does is package up that autosome and turn it off, right? It silences gene expression on that autosome and just inactivates it. And when these autosomes are inactivated, they tend to move into this region of the cell called the sex body where the inactivated sex chromosomes are during this meiotic stage. And so there's some link between these inactivated autosomes and the disruption of the inactivation of the X and the Y. And so this was a great model for how a mechanism for this overexpression of the X chromosome that we could actually test in our data, because what you would expect is the sterile male where the X and Y are disrupted should have autosomes that have divergent PRDM9 binding sites, and those autosomes should have lower expression in the sterile male because they should be inactivated, right, if they're disrupting this inactivation. And so what we did was we looked at the degree of how asymmetric that PRDM9 binding is with the expression in our sterile hybrids, and the chromosomes that had the most divergent PRDM9 binding sites had the lowest expression, and these are for gene windows along the autosomes of our hybrids. We saw lower expression for those chromosomes. And so this was really the first test of this hypothesis in an actual natural hybrid that we can confirm that there does seem to be something about PRDM9 binding and the disruption of the X in the sterile hybrids. So that's cool. So we got a mechanism. It looks like this X inactivation is associated with PRDM9, and this really makes sense given those earlier mapping studies I showed you where there seems to be that association between sterility genes on the X and that locus on chromosome 17. So then let's look at the last stage of development. This is the one I'm excited about. So here we see this really traumatic overexpression of the sex chromosomes in this post-meiotic stage. And I'm not sure, we weren't expecting this when we did this experiment. We thought all of the cool stuff would be up here at that PRDM9 part of the story. But this is really intriguing because this overexpression, it could just be a downstream effect of the earlier disruption, right? It could be you mess things up early, it screws everything up from then on. That's possible. But there's another hypothesis that this gene expression on the sex chromosomes at this stage, we know from other studies in mice, is associated with these two X and Y linked genes, SLX and SLY. So SLX and SLY are ampliconic, or multi-copy ampliconic genes that form these repeated regions on the sex chromosomes. So they have multiple copies across the sex chromosomes. And they appear to interact in this coevolutionary arms race. So if you take a mouse that has a normal balanced copy number of SLX and SLY, and you knock down the X version, so you have too many copies of the Y, you get slightly abnormal sperm morphology. You get lower expression of the sex chromosomes. You turn sex chromosome expression down, and you overtransmit Y-bearing sperm. So you have a sex ratio distortion. So that's what right there suggests there's some sort of antagonistic, selfish evolution going on here. If you do the opposite, if you knock down the Y and you have too many copies of the X, you get really abnormal sperm morphology. You overexpress the sex chromosomes, like we see in our hybrids, and you get a biased transmission of X-bearing sperm. If you knock down both copies, everything returns to normal. You get normal sperm, no sex ratio distortion, no overexpression of the sex chromosomes. So what this suggests is that these genes are undergoing basically an arms race where they're adding copy numbers. So remember, they're multi-copy genes. They're adding copy numbers in order to outcompete the other version and control that sex ratio distortion during spermatogenesis. And so this is a potential mechanism because what's really cool about these genes is they've undergone this massive expansion in copy number in the mouse radiation. So if you look at since these genes have evolved, they've gone from upwards of 180 copies in musculus, but only 50 copies in domesticus. So you can imagine if you make an F1 between a musculus and a domesticus, which is what we're doing, they're going to be imbalanced for the copy number of these genes. So you're going to have, here's our fertile F1, pretty fertile, and here's our sterile F1 is what those copy numbers would look like on the sex chromosomes. So our sterile F1 matches that hypothesis that the sex chromosomes would be overexpressed and have abnormal sperm in the version that has too many copies of the SLX. And so we wanted to look at this in our data, and so we compared gene expression on for our F1 hybrids relative to their parent chromosomes. So now we're comparing a musculus X in a hybrid with a musculus X, and a domesticus X in a hybrid with a domesticus X. And what we can see is the hybrids that have the fewer copies of the X, we see a slight underexpression of the X chromosome like we would predict in that model. And we see slightly abnormal sperm morphology. They're okay, they can still fertilize, but they're not fertile. They're not beautiful sperm. So where this one, the one that has the too many copies of the X, you see that really abnormal sperm morphology, the overexpression, and we see this when we compare the X to the parent chromosomes, but not when you look at the autosomes. So this is really good support that there could be something about this SLX-SLY model going on in our F1 hybrids, that it could be an antagonistic evolution of these ampliconic genes. Other evidence is that when we were doing this study, we looked at just gene expression across the autosomes in sliding windows across all the autosomes, and we found this one region on chromosome 14 that had overexpression in our sterile hybrids. This region on chromosome 14 also has a lot of paralog, a lot of ampliconic-looking genes that are non-annotated on the chromosome 14, and we thought, well, that's weird. That looks a lot like an X chromosome in terms of the repeats, and it seems to be overexpressed just like our X chromosome is. And right when we did this study, Julia Coquet's group came out with a study that showed that this region of chromosome 14 has the same epigenetic regulation as the X and the Y chromosome. So it suggests this region is being regulated somehow the same way as the X and Y chromosome during this developmental stage of spermatogenesis. So we think this is just more evidence that there is this copy number arms race going on between the X and the Y, and that this autosomal region on chromosome 14 might be associated or interacting with the X and the Y chromosome. So there's still that question then that I brought up earlier of, okay, how do we know though that this isn't just downstream disruption from this earlier stage? And so we could get at this. This is where we get back to that polymorphism. We can get at this when we look at, instead of comparing a sterile and a fertile hybrid, we could look at sterile hybrids. So here we're going to use different lines of mice, but we're going to be comparing mice that are sterile when you cross them this way and sterile when you cross them this way. And what we would predict is that we would see that meiotic disruption in both mice, but in this mouse, you're not going to have that SLX-SLY imbalance. You're not going to have that excess of the X, and so we don't expect to see the postmeiotic disruption in this cross. And so this is what those data look like. So this is just a reminder. Those are our F1s. That was the other cross. Here we're doing new lines of mice, and when you look at meiosis, we see that disrupted X inactivation in both reciprocal crosses. So it looks like the sterility in both crosses could be associated with PRDM9 early. But when we look late, we only see that massive overexpression of the X chromosome in the one that has the excess copies of SLX. So in the reciprocal cross where there's more copies of the Y, we don't see the overexpression, exactly like that model predicts. So we think this is really exciting evidence that SLX and SLY are contributing to this disruption of the X chromosome. Our next question is like, okay, but how do we link it to the phenotype? And so to get to that, we actually already have generated lines of mice that are a musculus mouse with a domesticus Y and a domesticus mouse with a musculus Y. And then we can look at the morphology of the sperm of those mice and actually be able to say whether mismatching the X and the Y has anything to do with sperm morphology. And so we're pretty far along, but then, and this has actually been done before for those of you who know their mouse literature, and the answer is yes, there seems to be some effect on morphology, but what we're doing next is we're going to do cell sorts now on these lines and be able to look at patterns of expression in each of these different lines. And we're pretty far along in this. We're already doing the cell sorting, but we're also getting to the point where we have enough generations where, remember, we're starting to see that skew in the sex ratio. So in the direction we would expect, we're starting to see over-transmission of the X chromosome in the ones that have the excess of X chromosome copies. And this is work that's being led by Emily Capagna in the Good Lab. And we're also collaborating with Peter Ellis to do a lot more detailed work on the sperm morphology that if you're interested in, it's a fun project. So overall, just to sum this part of the talk up, we see disrupted expression early. We see it associated with PRDM9 at meiosis. We see what looks like an independent mechanism in these postmeiotic cells that appear to be associated with antagonistic coevolution of the X and the Y. And so sterility, even though we tend to think of it as a single phenotype, it is complicated. There are a lot of different mechanisms contributing to hybrid sterility just in these crosses. And even though as a field we like to think everything is PRDM9 in mice, our results suggest that there are other factors contributing to sterility. And so really sterility is a composite phenotype. And so what I'd like to talk to you now, I think these results really highlight that sterility is not explained simply all by the musculus X chromosome, that clearly there's autosomal components. The fact that we have these sterile hybrids from some of the mice here suggests that those autosomal components are important. And so here's where I want to talk about a beyond PRDM9 component for sterility. And so we wanted to look at this polymorphism and really say what is the genetic basis and what might be some of the causes for this polymorphism. And so to do this, we basically took lines of mice. So these are actually some of the lines we used earlier. I was just trying not to muddle things by including nomenclature that no one will remember. But we're using these two lines of mice, PWK and CHEK2, which I'm just going to call purple and orange. And they're isolated from different regions, different allopatric regions. You will notice, though, that this purple one is fairly close to the hybrid zone. But these are allopatric lines of mice. But when you cross these mice with domesticus, you get, this is the one where when you cross it, you only get sterility in one direction, where this one, when you cross it with domesticus, you get sterility no matter which way you do the cross. And so here's where that asymmetry comes in. And so this is really powerful because what you can do is actually map hybrid sterility by taking advantage of this variation. So we took these two strains and we generated a musculus F1, so this is a within an intraspecific F1 that has DNA that is half PWK or half CHEK2, half orange or purple. And we did that in both directions. And we then test crossed those mice back to, getting ahead of myself, back to a domesticus mouse and generated a panel of F1 males that will either be sterile or fertile depending on which autosomal variant they inherit from these F1s. And so this is a way of us doing QTL mapping to look at mapping of these sterility traits. So we found 5 regions of the genome that, oh, it's really washed out, sorry. So these are, if you can see it on my screen, these are highlighted in purple because they contribute to sterility for the purple mouse, and these are highlighted in orange because they contribute to sterility. You can't see the rest of the QTL, but you can see the LOD scores, the peaks. But those are where we found genes that contribute to different sterility traits like sperm count, head morphology, testis weight on the autosomes of the mice. And so that was great. So sterility is really complex. We're finding multiple regions of the genome that are contributing to it. But what we got really interested in is this one region on chromosome 4. And that's because if you remember, right, chromosome 4 is the one where they're sterile in both directions. And in general, the orange mouse is the one where they're sterile in both directions. And in general, those are really sterile mice. Like when we do these crosses, so we kind of expected orange alleles to contribute to sterility. But in this case, on chromosome 4, these orange alleles, those were actually more sperm and better sperm in the orange mice. So there's actually this region where these orange mice had better looking sperm than the purple. And what's really cool about this region is if you look at chromosome 4, you see this huge 20 megabase chunk of chromosome 4 where this QTL corresponds to that has distorted transmission in our crosses. So instead of the 50-50 Mendelian segregation like you were taught in intro bio, there were 70% of the alleles were being inherited from the orange mouse. So there's something about this region that was over-transmitting in our crosses. And remember, this is a region where the mice had more better-looking sperm, and they're over-transmitting in the cross. And if we looked more carefully at this region, there were a lot of fixed differences between the purple and the orange mouse in this region. So for being the same species, there were way more SNPs in this region than you would have expected to find because they're the same species. And so we wanted to know where this variation came from. This is an interesting region. Where did this variation come from? So what we did was we used whole exome data for a bunch of different strains of Musculus and domesticus, and we looked at the regions that are outside of this distorting chunk, so the rest of chromosome 4. We looked at the regions inside, and what we found is that inside this distorted region, we see mice that should be domesticus falling in with the musculus, and we see mice that should be musculus falling in with the domesticus. So what we see is what looks like multiple introgression events from in both directions in this region. So it's really weird. So it suggests that this region somehow has like a propensity to introgress in the hybrid zone. It's also associated with sterility in the purple mouse, so better sperm in the orange mouse, and appears to be distorted on our crosses. So we're like, how does this variation, where is this drive, where is this variation coming from? So we wanted to look deeper in the phylogeny, see if this is something from some other mouse. And so now we expanded this out to whole exome data for 10 species of different mice. And these are just sorry, I didn't say this, but these are just D statistics in a stepping window across the chromosomes to look for patterns of introgression. And what we see is we don't see any weirdness here in this lineage, so this is something that's only going on within that musculus radiation. So within things that form a hybrid zone. So then we really wanted to pinpoint the timing of distortion in this cross. So this was the next step. We wanted to say, okay, it was distorting in our crosses. Is it because the sperm somehow swim better? There's more of them. There's producing more. What is it? And so the first thing we did was we just took those F1 mice and we sequenced whole pools of sperm from those F1s, and we looked at the frequency of the orange and purple alleles, expecting to recover that 30-70 skew that we found, and we didn't. We found 50-50. So there's no difference in the production of sperm that causes this transmission distortion. So we were like, okay, maybe it's got to do with something to do with like the female background, and so we took those F1 mice and we back-crossed them, but this time to a musculus. So we're not worrying about differences between species, we're just saying, okay, how do these orange sperm fertilize? Again, we saw 50-50. And I'd just like to point out for those of you who do work on vertebrates, that was a lot of mice for a negative result. And it was thanks to the very hard work of my undergraduate, Vanessa Stewart, that we raised almost 600 mice to find 50-50 segregation distortion in our mice. So what this it seemed disappointing at first, but I think ultimately it's more complicated and interesting to think about. This distortion takes place only in the context of the domesticus background. So this is an allele in musculus that when it's combined with a domesticus background, causes this transmission ratio distortion and is also associated with these fertility phenotypes. And so to me, if you took away the sterility QTL, if you just looked at it, just thinking about the distortion, it's hybrid inviability, right? It's some sort of incompatibility, Dobzhansky-Muller incompatibility that leads to some sort of early zygote death, and so you just see fewer of the purple alleles in combination with a domesticus background, and that makes sense, except that we also had that sterility thing. So how is inviability related to sterility? I don't honestly know, but what I think, and this is consistent with some work that's been found in Mimulus, I know plants, but like Dobzhansky-Muller incompatibility in Mimulus, it could just be that they're physically linked, that there's like a sterility QTL and an inviability QTL that are just close together in the genome, and so we're recovering them both. It's mice, our QTL regions are huge, right? It's like half of chromosome 4. So it could just be they're physically linked. It could be they're developmentally linked. It could be something about an early inviability then relating to how sperm are developed later in a hybrid. Unclear. But what I think is the coolest thing about this study is don't forget that this region was reciprocally introgressed in our different lines of mice. You don't expect a Dobzhansky-Muller incompatibility necessarily to introgress. Like, you think of it as a barrier to gene flow. They should be things that are fixed, like going back to the beginning of the talk, they should be fixed. They should be the things that keep the species separate. But what we're seeing here looks like this region actually has a propensity to introgress. And so we think that this is an interesting example where the incompatibility, once it does introgress, and there is a hybrid zone, so you can get introgression of alleles that might contribute to incompatibilities. And then once it's there, that's then going to cause the drive or the further incompatibility that can maintain it in a population. So we think there's an interesting complexity between like both gene flow of an incompatibility and then how it sticks around near or in a hybrid zone. And so just to finish, because I know you guys got to take off, it's the end of the day, we found that genetic basis of the autosomal sterility is complex. We found several QTL. It's highly polygenic. We found a region that suggests that at least one of these regions, that this polymorphism is maintained by this combination of gene flow and an incompatibility. And overall, I think all of this work just highlights the fact that in a hybrid zone like mice, and I think this is true for a lot of hybrid zones, that there is a lot of variation geographically in the hybrid zone. And to really begin to understand this, we have to do more studies. So I'm saying this even though I recognize I just gave a whole talk about inbred lines of mice that were just plucked from the hybrid zone and kept in the lab. But what I think the future of these studies are doing more work with the mice in the hybrid zone and starting to understand the genetic basis of these sorts of traits using actual mice from actual populations. And there is work that goes on in the mouse that is like that, and I just hope to see more of it in the mice and also in lots of other systems, because there's going to be a lot of these different combinations of factors that restrict gene flow. So with that, I would like to thank all of the people that were critical to this work. Jeff Good, I did this work as a postdoc in his lab. None of it could be done without him, and he's been a great collaborator, and we're continuing to work on these systems. And I'm always taking, I'm not always, but like if you're interested in joining the lab, please talk to me. I'm definitely recruiting. And thanks for your time. It has been a fabulous day, so many great conversations, and I look forward to tomorrow. So.
All right, thank you, Erica. We have time for some questions.
So is there any ecological factors driving the divergence of the two subspecies?
So there's been some evidence, there's some classic work that suggested maybe the hybrids have higher parasite loads in the hybrid zone, so like a type of hybrid inviability that is related to the environment. But those haven't been well replicated, so it's kind of unclear, but it's possible that the hybrids are just less fit in general, and so they have more parasites. In terms of their ecology, that could be, they're commensal with humans, and so they occupy really similar barns, houses. There's not a lot of ecological differences that I would think would be important in this hybrid zone, other than possibly the hybrids might not be very good in the field. Yeah.
great talk. So I'm curious about, I know that there's obligate pairing of the X and Y in the mouse, and is there anything known about the interaction or the disruption of pairing when you had this asymmetry with PRDM?
I don't think there is.
Very cool to look at.
It would be, and I feel like the XY, so what she's saying is basically the X and the Y, even though they're really different, they still pair over a small region of homology called the pseudo-autosomal region. And I don't think that anybody in the PRDM9 context has spent much time looking at whether there's disrupted pairing of the X and the Y.
Right, because that shuts down spermatogenesis.
Too. There's so many checkpoints in spermatogenesis. Yeah, that's a great question. The other thing that I always love to mention, just to complicate this story, is I talked all about the balance of the X and the Y and the imbalance in the hybrids. So a nice little mouse fact. So our classic line of mouse, that is the mouse reference genome, C57 black, it's a domesticus mouse, but it has a musculus Y chromosome. So it's just our model organism for mice is a chimera. Really, it's a hybrid, right? It's got the wrong Y. So what this means, so all those knockdown studies I showed you earlier were done in C57 black, the model mouse, and that Y shouldn't even be in there. And so I was talking to Jacob Mueller today, and there's very cool possibilities that these copy numbers are gained and lost so quickly that this can actually happen, he believes, observably in the lab. And so it's actually possible that the Y is okay in that classic mouse because they have readjusted copy number to be in balance. It's a possibility that I don't think anyone's actually tested. Anyway.
Are there any more questions? No? Okay. Thank you, Erica.
Thanks.